Hi there, it's me again. This is the one of the last videos that we're going to do on chapter 10. So in this video we're going to look at how the cell cycle is regulated and how that relates to cancer. And then in the end we're going to just take a real quick look at how bacterial cells and prokaryotic cells go through the process of cell reproduction. So for now, I'm just waving and saying hi. I'm going to get rid of me here in this recording just so that I'm not obstructing any of the slides. So here I go. I'm gone. All right. So when we look at the cell cycle, what we notice is that the cell has all sorts of little checkpoints, which are the moments where a cell decides whether or not it's ready to move on to the next step. And those checkpoints are regulated by internal signals and external signals. And so we're going to take a look at some of the ones that are really critical to a cell in making those decisions. So there are two kind of categories. One are positive regulators. Positive regulators are the ones, just like it kind of makes sense, because these are the ones that are going to stimulate or promote movement to the next part of the cell cycle. These proteins are actually coded, so most everything we're talking about again are proteins. So these proteins, these regulators that are positive regulators, are coded in an organism's DNA, right? So in that particular cell, there's DNA that have genes that we call proto-oncogenes. And these are ones that are going to make these positive regulators. So a couple of them that can act as positive regulators are things like growth hormones. So this makes sense, right? So growth hormones, which are at a higher level when the organism is small or younger and is growing. And then cyclins. Cyclins are proteins that are produced, and we're going to see a little graph of this coming up, and that can act as positive regulators of the cell cycle. Negative regulators would be the other way, right? These are the ones that are sort of the stop signs that say, no, we're not going to allow the cell to move on to the next cycle until certain things happen. So these are important because they're going to inhibit a cell from moving too quickly through the cell cycle. These proteins are also coded in the DNA by things called tumor suppressor genes. And if you think about it, that term probably makes sense because what we don't want are tumors. So these genes or these proteins, which are coded by these genes, serve to not allow cells to move too quickly through the cell cycle and therefore produce too many cells. And these proteins can be a number of them, but these are the three ones that we're going to look at. Something called RB, which actually stands for Retinoblastoma protein, which was discovered during looking at a condition called retinoblastoma, which is a type of cancer actually. P53 and P21. All of these are negative regulators. So let's start here with cyclins. So there are naturally in the cell things called Cdks. And Cdks I'll get into more detail in the next slide too. But Cdks are cyclin-dependent kinases. Now you've heard the word kinase before. We saw that when we were looking at cell respiration. So a kinase is something that generally has to do with activating or deactivating things using one of those phosphate groups. And then there are cyclins, and there's different types of cyclins or flavors of cyclins. They have cyclin D, E, A, and B on this diagram. And in order for CDK, these cyclin-dependent kinases, to become activated, they have to bind with a cyclin, and then they do their job from there. So let's take a look. So this is, again, positive regulators, right? So cyclin is going to be made by the cell in response to some signal. Once its level goes up, and it's going to start to bind to the Cdks that are there. And what it is going to do then is going to be able, so here's the kinase part, here's Cdk, and here's the phosphate. And so once that Cdk cyclin complex is formed, it's going to be able to take a phosphate group and put it onto some other protein. So here you see phosphate being added to a target protein. And by putting the phosphate on it or phosphorylating it, we activate it. We're going to activate this target protein, which is then going to go do something else. Then ultimately, when this is no longer needed, the cyclins get broken back down again by enzymes. So here's a view of this. Notice that we are right now primarily here talking about the G1 phase. So G1 has a low level of cyclin in the beginning when the cell has just gone through mitosis, still developing. But over time, when that cell has grown and developed and matured and probably done a lot of its job, the cyclin level starts to rise. And the cyclin level, cyclin D, rises. And then also you can see later in the G1 phase, you have cyclin E begins to be created in quite a large supply, and then cyclin A also. And so these different cyclins have different cycles where during the different phases of the cell, G1, S, G2, and then ultimately we get into mitosis, these levels rise and then fall again. In negative regulation, we have these three molecules, this retinoblastoma protein, RB, and then something called P53 and P21. And these three have activity at other parts of the cell cycle, but we're going to look at it primarily at this G1 checkpoint. So RB normally is, we can see it right here, RB is normally unphosphorylated. And when it is not phosphorylated, it's going to bind to what we call a transcription factor, which basically is going to be something that's going to stop any protein or a particular gene from being expressed. So this RB combined with this complex, which you don't need to worry about, but is going to arrest or stop a protein from being made in the DNA. But in response to some of the other activity we're going to be talking about, RB now can be phosphorylated. And what you see right here is that when an ATP is used and is going to phosphorylate this RB, and this is going to happen from those Cdk cyclins, what it does, the phosphorylation is going to release RB, and that from that transcription factor, this little thing here, which is then going to allow the cell to move forward. So in its normal state, as a negative regulator, RB is not phosphorylated and therefore is inhibiting the cell to move on and to go into the next, to the S phase. And here's just another diagram that I've kind of elaborated on here. So RB is going to monitor the cell size, and it's regulating when it's not phosphorylated. But when RB is phosphorylated, that disinhibits it. So it's inhibiting things happening here. It's not allowing the proteins to be made or the genes to be used here in the DNA to make those proteins until it has a Cdk cyclin that phosphorylates it and then disinhibits it, allows it to be released, and therefore the cell now can do what it needs to do to move to the S phase. All right, two other inhibitors, P53 and P21. Now, P53, again, is we're talking about in the G1 phase of cell cycle. It has a really important role, and what it does is able to detect if there's been any DNA damage. So if there is damage in the DNA, that cell got exposed to something that actually damaged the DNA, then p53 can do a couple things. One, it will see whether or not that damage can be replaced or repaired, and if it can't, it's going to generate this p21, which is going to inhibit the CDK cyclin. I know this is confusing, but I'll draw it out for you in class. So p53, when there's DNA damage, is going to increase. We're going to get more p53. That's going to create this other one, p21, and that's going to stop the cell from moving on in the cell cycle. Now, if p53 recognizes that there's no way to repair this DNA, then it can trigger an event that we're going to call apoptosis, or programmed cell death, and I'll explain. So programmed cell death is a way that the cell basically self-destructs. Sometimes it's called cell suicide, but it is a way of recognizing that there's something wrong with the cell. There's some DNA damage, and so p53 is going to ultimately, if the cell that DNA damage can't be repaired, it's going to go ahead and trigger some signals which activate another set of enzymes called capsases. And sorry, I said it wrong, caspases. So caspases they do all sorts of interesting things, but basically they're going to cause the cell membrane and some of the membranes in the cell to break down and to do what we call blebbing. It kind of forms these very characteristic look when the cell is basically self-destructing, and then will get recycled by the cells around it. So there's really two ways cells can die. One is necrosis. And necrosis is real damage where the cell is either starved for oxygen, not getting nutrients, something like this. And we associate this with disease. So necrosis, here's just a kind of a cartoony picture that shows the effect of a cell that is undergoing these kind of changes. And so the immune system is gonna kick in and come and try to attack really and get rid of the cell because there's serious damage. But as opposed to apoptosis, which is programmed cell death, where the cell is going to purposefully self-destruct. Now really there's a couple ways that apoptosis happens. It can happen when there's a problem with the DNA, just like we just described. It also can happen really early in development. So if you've seen pictures of a fetus where they have, and you had this too, sort of this webbing that can exist between the fingers and the toes, that we get rid of that mostly before we're born. Occasionally that doesn't happen. And so apoptosis is also a way of removing those cells that aren't going to be used once that organism is fully developed. Now, cancer, we're going to go through this in an interactive that I'm going to show you. But basically, something goes wrong with all of these different checkpoints and all these different regulators of the cell cycle. So cancer is uncontrolled cell growth. And when a gene is that normally codes and is healthy for either the positive or the negative cell regulators, if there's a mutation in those genes, then that can lead to the cell moving on in the cell cycle prematurely and fairly dramatically. If RB, remember this is the negative regulator, if it no longer can control the cell from moving into the S phase, then what you're going to get is uncontrolled cell division. So the brake is no longer applied. Now I think I'll wait here and talk a little bit more about cancer in the next video. So I guess we have one more to go. Here we go.